Ma'am, you can go now.
Good morning, everyone. I welcome you all on behalf of students, staff, and management of Bapatla College of Pharmacy and Bapatla Educational Society for this day 4 of 1 week online international faculty cum skill development program on innovations in drug delivery system organized by Department of Pharmaceutics, Bapatla College of Pharmacy in association with the Sri Padmavati Mahila University, Tirupati and IPA Education Division. I'm so pleased to have the chance to introduce our today's guest speaker, Dr. Amir Shirzai. Dr. Amir serves as an assistant professor of pharmaceutical sciences at Marshall B. Ketchum University College of Pharmacy. He holds both the PharmD and the PhD degrees. He has received his doctoral degree in pharmacy from Chapman University School of Pharmacy and PhD from the University of Rhode Island. During his PharmD studies, he had opportunities to work and perform rotations at CVS retail stores, Kaiser Permanente hospitals, and St. Jude Heritage Medical Group. During his PhD and postdoc time, he has served as a research fellow at Chapman University, University of California, Irvine Medical Center, and was an associate member of Child Family Comprehensive Cancer Center at University of California, Irvine Medical Center. His major research is focused on the application of cyclic peptide-based drug delivery systems for intracellular transporting of various small molecules, drugs, and large biomolecules. As a result of his research investigations, he was able to publish more than 60 research papers in prestigious journals in the field of pharmaceutical sciences, which have been cited extensively by his peers. We welcome you, sir. Dr. Amit, you can share your screen and start the presentation.
Thank you so much for the introduction. And I just need to make sure that everybody has the presentation. So everybody can see this?
Yes, sir.
Very good. So I'm just going to talk about, first of all, thank you for inviting me. That's a pleasure to talk. I'm like very young to speak among all the big people. So I appreciate your time and your interest. But what I'm going to talk about more about the aspects of cyclic peptides, which when we started working on this project, peptides were always important in any drug delivery system. A lot of like antibiotics are peptide based, but cyclic peptides never had that much of attention. So we started to explore more about the cyclic peptide. And what I have to say at the beginning is this, what I'm presenting is some sort of a part of a bigger story that has been done with more than, 30 people around me. So I was a part of this group and I was just the one who tried to continue this trend. And, I'm presenting like a work of so many people who collaborated with me and helped me in this journey. So we go over the cyclic peptides drug delivery systems. This is just something to show about the value of like drug delivery. When you talk about drug delivery in India, it's a big topic, but when you come to the US and you talk like to the pharmacists about drug delivery, they think like McKesson, which is a big company, it's like delivering drug. And the majority of them don't have a big picture of about how important is that. So that's why one of the areas in pharmaceutical sciences that has been almost the same kind of in like last, 2 decades, it's the drug delivery. They still use same system, tablets, capsules, and because like insurances have to pay for them, so it's cheaper to make them using older drug delivery systems. But nowadays, if you see more and more, especially drugs for oncology, they are trying to conjugate different drugs with antibodies, attaching like linker through a linker, and these are expensive drugs. So the importance of drug delivery is just as important as like any other topics in pharmaceutical sciences. And you always know there are multiple steps. That's how you predict where the medication would end up. What are the barriers the medication has to pass and be stable to get to the receptor and site of the action. So that's more of like what we call the like PK and PD, and everybody knows about like ADME profile of different drugs. What are we gonna do about drug delivery? What are the classic stuff? They can reduce the potency of the parent drug. They could be toxic. They have to be added to the cost of the final cost of the drug, and they can cause complications due to different reasons. So far has been, different tools have been used to improve the delivery efficiency of different types of drugs. Things like liposomes have been used like prevalently. I remember, I guess if I'm not wrong, 2014 or 2015, there is a liposome formulation of doxorubicin, which is an anti-cancer drug, and there was a shortage at hospitals in the US for Doxil because it shows a better profile in terms of cardiotoxicity in patients. So that's where it comes to the picture, how important is that? Same thing like surfactant, nanoparticles are becoming a big deal from the discovery now to the market. Antibodies are pretty much every year, if you're teaching or if you're reviewing literature about biopharmaceutical, you see, oh my God, every year you have to update your slides. There are so many new drugs. They're antibody based. They are like big molecules, and these are all the big companies who are coming to the picture to introducing a new era through personalized medicine. So that's how it's important. And viral vectors. You always need to know, it's actually very important if you're working in lab, or even if you're a pharmacist. If you're working in lab, you need to know what is the toxicity level, how much of the drug you want to be released in body, how much is the stability of this drug, what is the therapeutic level, and things like that. So essentially, we want to have a certain dose of the drug to be active inside the cells, but it shouldn't pass a certain dose to become toxic. It has to stay in a certain window between these two levels to be efficient but not toxic. So that's when the beauty or the art of like drug delivery design and different systems would come to the picture. And then how long you want to, you want the carrier to release the drug. One thing I want you to say, a hospital pharmacies here need to know the half-life of different types of drugs. Let's say patients come to the hospital, they take a medication, then they go home, they have side effects in like 7 days, and they come back and say, what's the problem? So essentially, it's a fast-paced process. So patients taking 2 or 3 medications every day. And the hospital pharmacy need to remember that 7 days ago, this patient took this medication, and this medication is still circulating in the body. So that's how it's important about the design and knowing the half-life, even if you're a hospital pharmacist. But what is the problem of like peptide-based delivery systems? Peptide, you don't see that much of peptides to become like bio-orally available. They're not resistant in the presence of different enzymes, especially peptidase, especially if they're like linear peptides. They have poor absorption, and then they're not very again stable. So the amide bond may be get degraded at any point in the body. And then the majority of my target was focused on cells. So I was not a biologist, but we had to have enough like essential knowledge about the structure of cells. Cells composed of different compartments, depends on how it gets to the proximity of the lipid bilayer. The drug delivery system has to carry the drug inside to the cell intracellularly, or they just dump them in the vicinity of the cell membrane. So there are like different mechanisms, and there are lysosomes. It can be degrading the drug, and you wouldn't see the effect. And then at the end of the day, if you're targeting the DNA, the gene machinery of the cells, you have to reach the nucleus, and nucleus is not an easy target. So that's the importance of how much we need to know about structure of the cell. So essentially, we need to know the lipid bilayer, which is a you know, a barrier. And this structure is like always you need to know that hydrophobicity and negatively charged elements in this structure of cell membrane could be a good target if you're designing a drug delivery system. So we picked some peptides. That's how we started our story. We wanted to make a library of cyclic peptides and see if they can, you know, deliver the different types of drugs into cells. So we pick like positively charged amino acids and negatively charged amino acids, and we combine them alternatively with the positive charge and negative charge, negatively charged amino acid and positively charged amino acid. So let's say hydrophobic and negatively charged, hydrophobic and positively charged, hydrophilic and positively charged. So that was like a randomized library, and we wanted to see what sequence actually works the best in terms of carrying the drug, transport the drug into the cell. So among all those synthesized peptides, I've been, I mean, working with these peptides for maybe more than 11 years, and uh, I don't know, I can tell you we make more than hundreds of peptides, and these are cyclic peptides, hard to make. The quantity is not that much. The purification is a hassle. So uh, many people say 100 peptides is not that hard, but when it comes to the purification with HPLC, and then you have to have enough quantity for all the assays, it's a lot of work. Uh, so among all those peptides, cyclic peptides, we found out that the sequence of arginine and tryptophan works the best in terms of delivering a fluorescence label anti-HIV drugs. This is F3TC lamivudine. It's a pretty old drug. The reason we picked this drug was like that was a good drug back in the day. However, this drug was not able to cross the cell membrane efficiently. So we used this delivery system to take this compound into the cell. And then we had this fluorescence marker to study using FACS of how much of the fluorescence signal we have inside the cell. So the higher the fluorescence signal, the better the carrier is. And we found out that the combination of arginine and tryptophan works the best among all the cyclic peptides. When we did microscopy, we found out that the target of these cyclic peptides is the nucleus. So they actually cross the cell membrane and cross the nuclear pore complexes and get to the cell membrane. We use that through DAPI microscopy. But when we look at transition electron microscope, we find out they form some sort of a structure. The structure was different in terms of morphology and in terms of size, diameter size. Depends on the media, depends on the time, depends on the, you know, what type of peptides we are using. So we found they have like formed some sort of a structure that shows some sort of self-assembly for these peptides. And we tried like cryo-TM, we tried like negatively staining TM, and it was very interesting to find out they have actual structures. For the surface, we use a scanning electron microscope. So that shows also the structure. And that's how we found out these peptides actually have some sort of nano-sized structures as it is. So the conclusion of this part of the work was we already know what is a good combo. The best combo is the presence of arginine, which is positive charge, and tryptophan, which induces hydrophobicity to the system. And they can work as nuclear targeting system. It was very interesting when we did the, you know, we wanted to see if this is how this system actually carry the drug into the cell. Is it through endocytosis? Is it through like different mechanisms? And it was not. It was like not energy dependent. So if you know, you just put the cells in 4-degree fridge and it shows you if the uptake is different at room temperature versus 4 degrees Celsius. If it is not, it means it's the temperature doesn't play any role. So that was like very interesting to know that this is not energy independent. So now I knew this peptide would work for small molecules such as 3TC. Now I just wanted to see if they actually can carry larger molecules into the cell or not. So we had somebody working in lab with phosphopeptides. These are the prototype for use for signal transduction. And I just said if you made the fluorescence label of these, phosphopeptides, which are big molecules, and they contain a phosphate group, which is negatively charged. So why not I try this and see if they can cross the cell membrane in the presence of the peptide versus when they are alone, because they don't cross the cell membrane easily. They are very big and they have negative charge. So that's hard to cross the cell membrane. So when I tested, I see, wow, this is essentially the phosphopeptide alone. This is the phosphopeptide alone. And these are in the presence of my cyclic peptide containing arginine and tryptophan, containing a hydrophobic amino acid and a positively charged amino acid. So the uptake is increased significantly in the presence of these peptides, showing that this peptide is able to carry larger size molecular cargoes into the cell. So I found very cool. Let's look at the structure. It was also confirmed with microscope. But then circular diachrosm was something that we used to see the orientation of amino acids. I wanted to see if they actually bind and the orientation of amino acids would be changed in the secondary structure of the peptide. So we did the circular diachrosm, and we found out upon mixing, they actually has different structure. So that was something interesting for me, that the amino acids, the charge, electrostatic charges, and the hydrophobicity portion of that carrier is also involved in terms of interaction here. Now, I wanted to see under transition electron microscope that this is my phosphopeptide, this is my cargo, and this is my peptide, this is my delivery system. When I mix them together, it's just water at room temperature, 1 hour incubation. It's nothing like a special procedure. But then when I take a picture of like under transition electron microscope, I see a different structure. So they're actually binding. They form some sort of different morphology, showing that there is interaction among these peptides and carrier systems. So that was very interesting to me, that they have different, morphology inside. So the conclusion was like these cyclic peptides containing positively charged arginine and hydrophobic tryptophans can work also for the large size molecules. And then we have some sort of self-assembly process, which we worked on them for like 3 years, but we couldn't actually figure out how to engineer that self-assembly aspect of these cyclic peptides. We just draw like a library of different characteristics that can be different in terms of morphology. When there is positive charge, this is the structure. When it's negative charge, it's the structure. The size is different. So that was like, we didn't go through the how to make them prepare for our tool, but we kind of characterize their features here. So back in the day, it was very hard to do gold nanoparticles left and right. People were doing like gold nanoparticle, and I had a friend who was working on gold nanoparticles. I just wanted to see if this system can actually work with gold nanoparticles, or they can help the delivery of drugs in the presence of gold nanoparticles. So I tried to mix them together. There was a gold solution and there was a peptide solution in water. So essentially, the classic procedure is you go through the reduction. You reduce the gold from AU3 to AU0, and that's when gold becomes nanoparticles. So the color changes from yellow to between like pink or like purple type, but depends. So I said, let's not do that. Let's just mix them together, because there was a paper showing that tryptophan can actually reduce gold to gold nanoparticles. So I just wanted to see if I can use this cyclic peptide as a reducing agent at the same time delivery system. So I just mixed them together, and maybe after like 5 minutes, immediately you see the difference in color. It's actually turned to purple or more like a pinkish kind of color. So that's how I find out that this peptide is actually able to reduce the gold to gold nanoparticles, at the same time cap the gold nanoparticles. So we have the size of the peptides, kind of fixed when we mix them with the peptide. Again, it was beautiful. We ran the study for a whole library members, and among all those members, only a combination of arginine and tryptophan showed to be potent in terms of generating gold nanoparticles. Now I have a beautiful particle, it's capped, it's wrapped with peptide, it's reduced. What's the use of it? I just wanted to see if it's working or not. And that was the TEM picture of gold nanoparticle. They're a little big, but still, we were able to manage the concentration and come up with different, size and different shapes, so things like that. One thing to know is that this peptide was not that toxic compared to other peptides. So the cyclic peptides were not, I don't know, toxic up to like 100, 200 micromolar, significant, maybe 2% of cell death in the presence of these peptides. But it was a good tool. So I said, if it's not toxic up to 100, I'll just use 25 micromolar and I should be good. So that was one advantage of peptides that, I had versus like different types of carriers, even lipofectomy. So I did the same study with the fluorescence labeled drug, and I found out in the presence of peptide-based gold nanoparticles, I have higher cellular uptake. So this is the drug alone. This is when I mix the drug with the peptide. It's increasing compared to the drug alone. So peptide is actually carrying the drug into the cell. But when I have this combo of gold nanoparticle capped with the peptide, it's actually working the best, better than even the peptide alone. So that was very interesting. Wow, this is even more in terms of quantitative assay. If you have any question about this assay, this is flow cytometry. We just measure the signal of the fluorescence inside the cell. So that's pretty simple. You just incubate the cells with the drug in the presence and in the absence of your carrier, and then you measure it quantitatively. You say, okay, in the presence of the carrier, this is the number. In the absence of the carrier, this is the number. That's how you compare the assay quantitatively. And also, I just wanted to see, now that I have, gold nanoparticles and peptides, am I damaging the cell membrane integrity? Am I having like some sort of carpet type mechanism of internalization? Am I damaging it? So I tried to figure out if the membrane integrity is intact or damaged. And it was very interesting that the membrane integrity, even after the incubation, was intact. So that was a good tool for me to show that I don't know what's the mechanism of the internalization. So conclusion of this part was that formation of gold nanoparticle improved the transporting of the drug into the cell with the carrier. Also, that was a new method for me. It was like in chemistry called in situ, which is like everything in one pot. That was the same thing for me. I didn't have to make gold nanoparticles and then functionalize them with the peptide. So that was one chemistry type innovation for me. Now I have the cyclic peptides. I know it's working. Now I just wanted to make a similar linear peptide and see if they behave differently. If this is cyclic peptide and linear peptide, would they form different structures? Would they show different, behaviors? And so that was the whole idea. Let's say I mix the gold solution with cyclic peptides and linear peptide and see if they have different morphology, different size, different ability to transport the drug into the cell. And very interesting. So I made this KW, which is a lysine and tryptophan combination. There are like 10 amino acids, 5 arginine and 5 lysine. And when I say the previous one was like WR, so that's like 5 arginine, 5 tryptophan. This is like tryptophan, arginine in the sequence alternatively. So that's how we design this system. So now I just wanted to see if there is any difference between linear peptide and cyclic peptides. And very interesting. So when I had the linear peptide, I had this ball-shaped structure, which I had a nice cavities here showing there is a hydrophobicity repulsion in the structure of these peptides, in the structure of these gold nanoparticles. And when I had these cyclic peptides, the structure was totally different. So this is the linear peptide, gold nanoparticle. This is the cyclic peptide cap gold nanoparticle. So that's 2 separate structures. I can't believe that the presence of like the orientation of amino acids in the secondary structure of the peptide is making this huge difference for me. So that was very interesting for me to find a difference. But up to now, it's beautiful, it's working. I know it's a good deal. I just need to know what's the use of it. In pharmacy school at pharmaceutical sciences, it's not like chemistry lab. So you have to have application, you have to show it works. Otherwise, it's just a simple publication. It doesn't go that much. So I found a cell line that this cell line is resistant to doxorubicin, which was a anti-cancer drug. Doxorubicin is a very good drug. It's been in the market for so many years. It does have like a cumulative toxicity. So if patient receive after certain amount of the dose in the whole lifetime, they would be at high risk of cardiotoxicity. So what do we want to do? We want to inject as low as possible. We want to, keep the dose low. So we attach doxorubicin with these cyclic peptides through a linker. And then I just needed to study if that's making a difference or not compared to doxorubicin alone. At the same time, this is doxorubicin. It has like 3 different mechanisms, very good drug. And this is the synthesis of the peptide, just some chemistry for how do we make them. But what did I do? We made the 2 different prodrugs. The first one was doxorubicin attached to the cyclic peptide. The second one was doxorubicin attached to linear peptide. So I just wanted to see how would this prodrug beat the resistance of the cells. The resistance is important because the PGP pumps on the surface of the cells are responsible to pump the drug out. Let's say if we incubate the cell with the drug, after 1 hour, you have a certain dose, and after 1 hour, the pumps start pumping out the drug. So essentially, after 24 hours, you lose a lot of dose. So I wanted to see if I can, design something that actually beat that, efflux effect. So what did I do? I incubate the cells, SKOV3 cells, which is ovarian cancer cells, in the presence of doxorubicin alone. This is doxorubicin alone. This is just a control. This DMSO is a control. So this is the drug alone. This is a physical mixture of the linear peptide and cyclic peptide. So for doxorubicin, it didn't show that much of difference because doxorubicin has a very good cellular uptake. If you work with it, you incubate for 20 minutes, it goes like a bullet to the cell nucleus. However, when I have a covalent conjugate between doxorubicin and the cyclic peptides, I can see that I would retain a higher dose of the drug inside the cell. The same thing for the linear one, however, is not as good as the cyclic peptide. So that was a finding with microscope as well, that if I leave the cells with the prodrug, it gets into the cell, I let them incubate. After 1 hour, I wash out everything. I replace the media with the fresh media. There is no more treatment. So now in the fresh media, doxorubicin alone is pumping out the drug. However, this prodrug gets stuck inside the cell, and the PGP pumps don't pump out the drug. Therefore, we are beating the efflux effect. So that's how we came up with 3-fold higher dose compared to doxorubicin alone. And then we kind of saw a relative to up to 15% better in terms of cytotoxicity and anti-proliferative activity of this prodrug versus the doxorubicin alone towards time. Even when we increase time, you see bigger difference because doxorubicin is already out, but your prodrug is converting to the original drug. And then we did some HPLC to figure out, how long does it take to have like 99% of the prodrug converted to the drug. And it was almost like 72 hours of incubation. So the conclusion shows that this system, the cyclic peptides, is able to deliver the drug into the cell, to the nucleus. At the same time, if you make a covalent conjugate, there is a good chance that we beat the resistance for ovarian cancer cells. And then this system shows a 3.3 to 3.6 folder higher cellular uptake compared to doxorubicin alone. So that was some findings for us. Now, I just wanted to see if we can design a system that targets something in the body of like animal, in the in vivo studies. So as you know, cancer cells are hungry, they are fast. I don't know, if you put it in one petri dish, one normal cell, one cancer cells, you come back after 24 hours, you see the cancer cells are growing way faster. That's why their temperature is higher. That's why their pH is higher. That's why they overexpress folate. So the overexpression of folate is a target for so many targeting delivery systems that has been introduced before. So I just got the idea from the literature, and I attached the folate to the cyclic peptides through a linker. I wanted to see if we can have a better, circulation in the animal system that, it stays in the body for a longer period of time. This technique used actually in clinic for so many drugs, especially if patients have some sort of, they need to um, they need to inject some sort of drug that stays in the blood for at least 2 weeks. They add this PEG, and the majority of this administration are done at doctor's office. So it's not at home or any pharmacy. It's mostly doctor's office, and they give the patient like something like, I don't know, acetaminophen, Advil, or something to reduce the inflammation at the site of injection. So here I added this PEG to, improve the half-life of this system, and then C5.5 to be able to track the system, because I wanted to see where does this system end up accumulating. So we designed a mouse, and it was like two types of tumor we developed. One was HeLa cells, one was HEC293. These are normal kidney cells, very easy to work with. That's why I picked them. And one of them overexpressed the folate, and then the other one doesn't. So the HeLa does overexpress the folate, and that was the whole goal of this process. And then we start injecting the the peptide to there is no drug here. I just wanted to see what is the fate of this delivery system. So what happened is we injected 1 milligram of the compound, and then as you can see, there was higher accumulation of the system in HeLa tumors versus HEC293. And at the same time, after 48 hours of injection, the mice was sacrificed, and we took different parts of the mice, and we wanted to see where do we see more of the intensity of the drug. Compared to the the tumor of HEC293, we had higher intensity in HeLa. At the same time, we had a good like intensity of the drug in HeLa tumor versus different organs of the mice. So that's just a preliminary study. It's not done in like 300 mice. It was just like 3 to see if it's working or not. So that's this portion has been like under investigating, and we're working on it, and we are hoping to improve the system. So that's just something I try to do like in vivo. So one thing I wanted to say was we use these cyclic peptides for different purposes. They show to be good nuclear targeting molecular transporters for small molecules, for large size molecules. We further tested these peptides for delivery of siRNA. It was successful. However, versus lipofectamine, we didn't have that much of like the release. So when they tested, the uptake was better, but the release was not that good versus lipofectamine. We're still optimizing that system. We also use that for prodrug. So if we attach it as a covalent link to the cyclic peptides, what happened is you can beat the resistance through shutdown PGP pumps in resistant cells, and then you are trying to reduce the effect of efflux in this type of cancer cells. One fun fact that these cyclic peptides also used for SAR kinase inhibition, which is like some sort of now in oncology courses, they start teaching SAR kinase inhibition. These are the drugs, very easy, small molecules. However, these peptides were large molecules. We wanted to see if they actually inhibit the kinase or not. And then they showed a good potency, but we still haven't optimized the best structure for that purpose. At the same time, we use them as surfactant and stabilizing agent for different materials. Gold nanoparticle, I also use that for different application, using gold nanoparticle for delivery of siRNA, delivery of anti-HIV drugs, delivery of anti-cancer drugs. So we have done a lot of work in terms of seeing if this peptide can, make gold nanoparticles in certain size, if they can be in different morphology, if this is all triangular. And that's been very interesting. After that, I did selenium nanoparticles with these cyclic peptides, the same structure, and it was like a very nice orange type solution that shows this is a selenium nanoparticle with similar activity. And it shows that selenium nanoparticle also improved the efficacy of these cyclic peptides to transport the drug into the cell. At the same time, we use them for negatively charged phospholipeptides. Phospholipeptides were hard to, get into the cell. They are large molecules. They contain, negatively charged phosphate groups, so they're not going to cross the cell membrane. However, in the presence of these cyclic peptides, they form that complex that I showed you in TM, and they are able to deliver them into the cell. We also use these cyclic peptides containing arginine and tryptophan to deliver a lot of siRNA studies. And I still do a lot of work with siRNA to see if they can actually work, because, siRNA is large molecule and it's also negative charge, but then it's negatively charged molecule, when binds with the positively charged amino acids in the structure of the cyclic peptides, the release becomes a challenge. So that portion, we're trying to figure out what's the fine balance between hydrophobicity and the electrostatic interactions, and then when is this release has to happen. Recently, I've started working with the gadolinium nanoparticles. We're trying to see if these cyclic peptides are able to combine with gadolinium nanoparticles, if they can generate gadolinium nanoparticles, and then if they can transport the drug into the cell. In this case, gadolinium particles have been used for imaging for so long. They have a unique chemistry, and due to the presence of the electron in their orbital, they are being used for imaging prevalently, so much has been done. Even if when you go to MRI, you get an order from the doctor, it's written with and without contrast, to get a better picture to the doctor to see if they are missing something. So without contrast means without the gadolinium injection, but with contrast means when they inject you the gadolinium, and that's when they do the MRI. So I just want to see if I can use the gadolinium nanoparticle peptide cap delivery system to do the imaging at the same time the delivery of the drug into the cell or not. This has been under investigation, and we hopefully we get something to present very soon. In addition to doxorubicin, we also use this system for camptothecin. It shows similar effect. However, the camptothecin release was harder, the structure was harder. So doxorubicin is a smaller molecule compared to camptothecin. But then I need to make sure that this is it when I make the covalent bind between cyclic peptide and the drug, is this the prodrug converting to the drug inside the cell, or is just the whole thing is working as a new drug? So that's the portion that I'm trying to figure out. They need more studies to understand the use of the conversion of prodrug to the drug alone. And then that's the recent portion of my work. And again, the in vivo studies also have been undergoing. It's just very hard to stabilize a certain protocol, IRB, and all those stuff to get it done. I need to say thank you to everybody, to my previous boss. He has done everything for me, and I still is his biggest collaborator. Marshall B. Ketchum University, Chapman University, UC Irvine Medical Center, University of Rhode Island, and all those collaborators around the world. They helped me a lot, and everybody will be part of this journey. Also, I want to thank you, Dr. Rajesh Bapatla, to give me this opportunity. At the same time, Bapatla College of Pharmacy for giving me this chance to present my work. This is just a taste of cyclic peptides, and if you want to know more, just email me. I would be happy to answer any questions that you might have.
Very useful presentation.
Thank you.
Sir, there is a question from the participant. Vidyavadi has asked, what is the advantage of cyclic peptides as a transporter other than better entrapment?
So that's actually one of the questions that they have on the mini test, but I'll answer this question just to clear out for everybody. When we inject something into the body, the fate of that compound is very hard to predict. So we do different types of biophysical studies, we do different types of analytical studies to figure out what's going to happen, but nobody knows. So many drugs come to the end of the marketing aspect, and they just don't work. So when we use cyclic peptides, that portion of stability of these peptides compared to linear peptides plays a very important role. So they are not susceptible to the peptides, to the enzymes. If we want to, I don't know, get rid of if we want to make a compound more stable, we either change the amino acid from L amino acid to D amino acid, because they are like natural amino acids, can be recognized by the enzyme, so they get chopped. But if we use D amino acids, they don't. So that's the same strategy with cyclic peptides. I want them to be stable in the body, so I use cyclic peptides. So there is no carboxyl end or nitro amine end at the end available for the enzyme to recognize and start cleaving the peptide. So the stability is the most important aspect of this that I know.
Sir, is there any drawbacks with this cyclic peptides?
So as I said, when we combine arginine and tryptophan, arginine is positively charged amino acid, and it kind of induces toxicity. However, when tryptophan kicks in with the hydrophobicity, kind of reduce the toxicity of arginine somehow. The presence of guanidine group is actually very important for internalization of the, internalization into the cell through electrostatic interaction with the negatively charged phosphate groups in the structure of the cell, lipid bilayer of cell membrane. But what is the problem is this drug goes to the cell like bullet, sits in the nucleus, but the release is my biggest issue. That when this, let's say if I'm delivering siRNA, I want that siRNA to leave this peptide at some point and then do the job, the knockdown. But the problem with this peptide is they hold the siRNA, they sit inside the cell, and they doesn't let the siRNA to go away and do the job. So that's the biggest issue I have with these cyclic peptides.
Sir, the next thing is, it's not more of a question, it is a appreciation, because you have taken many of your studies and then you described it and then you said what is the outcome of all those studies. So there are more appreciations from the audiences regarding that. And is there any advice from your side for those master students who are doing research in this particular area?
Yeah, so I mean, I'm not ready to give you advice or give you any directions. It's just you do your best to to come up with the best outcomes, but always look up to the highest quality literature, because if I want to do something, if I want to repeat myself and look to my coworker and say I'm going to do the same thing, I'm going to publish a paper, I'll be graduating, who cares, that's something that you may say to yourself, but in long term, a project has to go for 5 years to 10 years, and it's always good to look up to like good literature, read like big journals, just try to make sense out of what you think that project could, work in benefits to you. Like I said, I am nobody to advise you, but I always, even if I'm not going to publish maybe in that journal, or I don't understand that aspect, but I'm just going to read it and see how it can be helpful to me. So I just improve the quality of my reading, and it gives me a better vision, in terms of interpretation.
Thanks a lot, sir. It was a very useful as well as a very cool presentation.
Thank you so much. I appreciate it.
On behalf of the participants and on behalf of the College of Pharmacy, we thank you, sir.
Thank you. Thank you.